more than one hat. I only thought about that. I could have brought a few hats from Asia to switch over while I started, but I didn't think of it until just now. So for the purposes of this, you know, Eric and I are going to divide up the time. I'm going to, although MedGenome's hosting, I'm presenting on behalf of GenomeAsia. So GenomeAsia is a nonprofit consortium. It's a consortium effort which is, you know, loosely organized, and that's partly just because of, you know, how it all came together. It came together with a variety of individuals recognizing that there was a deficit in studies that were being carried out in Asians. You know, there's a growing number of studies that are being done on East Asians, China, obviously Japan, and increasingly South Korea as well. But Asia is a very large and diverse continent, and there were really a lot of areas where there weren't the tools and the ability to carry out genetics. So what's on the right-hand side of the screen, obviously, was what we called the pilot project, published on the cover of Nature the end of last year. I'm changing the nomenclature a little bit in this talk. I'm calling that phase 1. And that's to kind of describe the trajectory of the efforts, and that I'll describe in the next slide. But then the bulk of it is really talking about this phase 2, where we're pulling together the findings, et cetera, and we're finalizing a manuscript for publication. It's important to say that, you know, as I said, it's a large, loose consortium. The MedGenome was a pioneering or founding member of that consortium. The other members, kind of core members, were and are Macrogen, which operates sequencing out of South Korea and the US, and then
Andy cut out.
Oops. You might have to finish the sentence.
Maybe I'll just text him and see if there's.
Uh,
He may not know, actually.
This hasn't happened before. Always.
The other thing is, I could jump in if he's not able to.
Yeah.
Get back in here. I just texted him here, so
right.
Let's see. Or maybe he just stopped presenting.
Does somebody else have the slides maybe that we could?
Eric, you wanna?
Yeah, you know, we can just switch, and then I can. Yeah. Why don't I present, and then Andy can jump back in at the end. Yep. So let me just share my screen here. 1 second here. Pull them up. And make sure I can find it here.
This is immensely awkward.
I can't find my screen on the. I guess I can do desktop here, but. Yeah. Okay, here it is. Okay.
Okay.
What's that?
All right.
Okay. Yeah, so sorry for the technical difficulties. Yeah, so mostly I'll be talking about SARGAM, which is our South Asian Research Genotyping Array by MedGenome. And Fall kind of hit on this, but basically we're a large diagnostics provider in India. We're sort of like the Invitae of India, offering a lot of different NGS-based diagnostics. And we have a research services arm in the US. And we're, as mentioned by Andy, we're a founding member of the GenomeAsia project. So really on the diagnostic side, through the diagnostic side in India, we've built a large network actually So we have a lot of clinical collaborators as well as different like in-hospital labs that are staffed by MedGenome. And so we have access to many different disease areas across India. And there's some really unique attributes to the population that I think Andy will touch on during his presentation actually. But yeah, so we've established this.
Andy's back if you want to.
Andy, are you back?
I am back, and I think I figured out why I disappeared, but there's no guarantees. But I don't want to interrupt the flow. If it makes sense for you to continue, I don't know how far you. It looks like
I just started, and why don't you start because it kind of sets up the why medical genetics in South Asia. Yeah. So. Yeah.
Sure. Okay, and hopefully. So what I know, where I remember that I was just highlighting the fact that it was a consortium with a number of contributors, including Genentech. And what I forgot to say, but I think it's obvious, is although this is a bioinformatics meetup, I am not a bioinformatics data scientist, mathematician, or anything related. I'm a molecular biologist. As I said, we published that pilot project that was really characterizing diversity across Asia. And then, as I said, renamed it Phase 1. So putting in that context, what's described here as Phase 1, Pan-Asian framework. So 2 goals, just kind of sampling genetic diversity across the continent, and then using those efforts to develop strategy for regional efforts. And that project was all based on 30x whole genome sequencing data. I'm only going to talk about Phase 2 efforts in South Asia. So when it says strategy for regional efforts, that's all I'm going to talk about. But if there's questions, etc., I'm happy to talk about other aspects or other areas of the continent. Again, the Phase 2 has been based on 30x whole genome sequence. It's different from the pilot or the project in that Phase 1, since it was sampling genetic diversity, it was characterizing isolated population groups. So for Phase 2, the sampling strategy shifted to sampling the general medically relevant population. So the individuals were recruited from the healthcare delivery system. So the goal then was to define the population structure with respect to its relevance for genetic studies, and then to develop tools for doing genetics, an optimized reference imputation panel and an optimized SNP array. So I'm not going to talk about those last 2. I'm really going to focus just on the population structure, and Eric's going to talk about those last 2. And I should say that the SNP array development has been done in collaboration with Thermo Fisher, and I know we have some Thermo Fisher folks on the line as well, so it's good to have them here. And then Phase 2 really builds the ability to do what I said, which is to increase the ability to do genetic studies or facilitate the ability to do genetic studies, in this case in South Asians. So what it enables then is the generation of a large-scale genotype phenotype data set along the lines of the Taiwan Precision Medicine Initiative, the UK Biobank, FinGen. There's a million veterans. There's a number of projects like this that Asia has basically been left behind. All of these, but now I think the tools are in place that really South Asia in particular is ready to join that same sort of effort. So why is it of interest? There's a number of reasons, and I know that many of you are aware of many of the reasons for why it's important to study genetics in populations other than Europeans. But I just want to highlight some specific factors that are related to the population structure rather than, just to a specific, kind of functional problem. So the origins of South Asia have some relationship to the origins of Europeans, and that's shown, on the map here from a paper by out of David Reich's group. So I'm not going to go into the details on that population history, but they have origins which are related to Europeans, but they also have origins which are very distinct from Europeans or from East Asians, such that, you know, tens of thousands of years ago, variants were accruing in South Asia that were not accruing in the same sense in Europe or East Asia. There's a very unique characteristic of South Asia at present is that there are thousands of distinct population groups that have maintained their identity. So there are literally about 6,000 that are recognized by the government of India. And what has been recognized from doing studies on isolated population groups, a lot of which has been done with SNP arrays, is that there are strong founder effects. And so I'm going to get into that, so I'm not going to say a lot more about it now. And that also what's apparent is, culturally apparent, is that there's high rates of endogamy, of, you know, marriage within the group, reproductive choice within a self-identified group, and then also of consanguinity. So I'll talk about that as well. So as I said, the samples that we used for this study were recruited in the healthcare delivery system. They were not recruited with respect to who they were. They were recruited in Pakistan by Danish Salehin in his efforts, the PROMISE study and the Pakistan Genomic Resource Study. So he's a collaborator in all of this. John Dinesh's group at Cambridge recruited samples from, and I should say those are largely cardiovascular in terms of cardiovascular hospitals, but all recruited in hospital systems without asking people, you know, what their group identity was. John Dinesh at Cambridge recruited folks in Dhaka in Bangladesh, and then samples were recruited in India, in West Bengal, through a general population study, and then in South India through a diabetes hospital, an eye hospital, and a muscular dystrophy diagnostic study group. So an important point is that samples were recruited from the three corners of the triangle that is the subcontinent. And if we just quickly go back, that is relevant to the origins of the population structure, the population history. There's a South Indian component that's been recognized, a North Indian component, and then there's also an influx from the east of Austroasiatic peoples. So those three corners that are geographically defined also correspond to where population migrations have occurred historically. So this is in the middle. So the left-hand side obviously shows where the samples came from. The middle shows a UMAP plot that FA did to look at the population structure of these various data sets. They're kind of color coded gray, blue, and whatever that color is, yellowish, greenish. So you can see, and I should say the UMAP was done with the global samples from phase 1 as well as these phase 2 samples, but I'm only showing you the phase 2 samples here in this plot. But you can see the clustering, of course, did include information from those phase 1 samples. And so you can see there's a lot of substructure within each of these three population groups, and that these three population groups do segregate, but with overlaps. And importantly, because we have those phase 1 samples, we can infer a lot of the population substructure. So I'm not going to claim that we're identifying individuals, but we can understand some really specific aspects of the population substructure that we see. So I'll just highlight, there's a black tail coming down the bottom in the center there on the right-hand side. That's a group de Lambada. And they were a group that historically were in the Northwest. They were moved to the Northeast, and now they're found throughout India, but they've maintained apparently their group identity, even though some of them are Muslim and some of them are Hindu, but nonetheless, they've maintained their group identity, even though they've been moved around, et cetera. So you see them there in samples collected in Pakistan. They also show up, I gotta get rid of my video window here. Sorry, I apologize. There we go. I can't see my own slides. I just recreated my problem. You can see it on the in the South Indian samples where we didn't have, as I said, the samples themselves didn't have group identity, but we can infer where the Brahmins are within the substructure, where the lower caste groups are found within the substructure. But then you can also see that Lambada group shows up again, or at least individuals which co-cluster with them. But then there's also questions that one would ask. These samples in South India were recruited from a major urban center. So as I said, these are samples, major urban centers, I should say. And India, like anywhere else in the world, individuals move into the cities, obviously, and they could move from anywhere, depending on where you are, from anywhere in the country, anywhere in the region, anywhere in the world. So most of the individuals in the South Indian group seem to be from South India. The Lambada we can explain in terms of historical migration, but there are a minority of samples that appear to be recent migration. So there's some Sikhs labeled with a pink KHA, Khatri, inferred to be Sikhs. There's Gujaratis and there's Bengalis, for example, but those are all minor. The bulk of the people who are coming to the hospitals in South India are South Indian. And then this is showing the substructure within the samples in West Bengal and Bangladesh. In this case, it's not the phase 1 samples that I'm layering on top to label the substructure, but it's actually the self-reported group identity for these individuals. So in this case, it was a community-based epidemiologic study. So we have a lot of demographic information on these specific samples, including self-reported identity, group identity, and then a number of other things that we can use in the analysis. So although I'm not illustrating it here, it actually validates the inference approach that we were using in other parts of the country. So for instance, the Brahmins shown here are labeled by the same samples. They co-cluster with the samples from phase 1, which were self-reported Brahmins as well. So, and there's other specific examples there. So again, I gotta get my cursor back. Okay, there we go. What we then did, and again, this is Fa's work, was look at the admixture. And so this illustrates what one of the points I was talking about in terms of the population structure overall, in terms of historical population migrations. So admixture, the best fit is with 4 components here. You can see, a yellow group on the outside, the Kolu on the left side, and then a green group on the right side. The buy-in, they're fairly pure. There's shared components with a number of the other groups, but they're minor. The major components from all these other groups are this blue and the orange. And so again, on the right-hand side, there's a group that's predominantly orange, the Santal. The Santal are an Austroasiatic tribe that still maintains their tribal lifestyle, tribal identity, etc. They've acquired agriculture about in the last 100 years. Traditionally, they were hunter-gatherers. But then you can see there's an Austroasiatic component to many of the genomes. And obviously, there's some arrangement here that's showing decreasing amounts of Austroasiatic and increasing amounts of the blue component, which is reasonably pure in the Brahmins. And from what we know about the population history of India, that's probably primarily what we would call a ancestral North Indian. So in this case, we've got, North Indian and Austroasiatic together with some groups which have had a very long history of reproductive isolation that are not admixed. And we don't really see an obvious component from South India, although it's probably in there. So again, that illustrates, again, the different, several points. One, the different components that contributed to the population, to the genomes from historical admixture events, but also the fact that these are maintained to some degree within individual groups. So there's been admixture, and then there's been some freezing of the mixture due to reproductive isolation within the groups. So I'm going to shift gears and talk about aspects relevant to both founder effects and consanguinity and endogamy. So I think it's probably intuitive that those are related. I mean, the term we use, founder effect, is obviously a description of genetic drift that we think about, or is named because of an effect where there's only a few founders contributing, a few individuals which contribute to the majority of the population that exists today. The most intuitive, semantically intuitive, is somewhere like Iceland, or perhaps Finland. Iceland's a little bit easier, 10 Scandinavian guys, 10 Irish women alone on an island for 1,000 years, and you have those founder effects. So there's no evidence for those kinds of geographic isolation in India. It's a pretty, geographically or in South Asia, I should say, it's pretty wide open. But there are cultural habits of reproductive isolation which can create some of the same types of genetic drift. So what's shown here is the level of homozygosity found in the 3 different population groups that I described. So there's, a couple of thousand individuals almost in each of these groups. And then we have a group of individuals from Taiwan that we're using as a reference group for comparison purposes. And what's plotted on the x-axis is the observed degree of homozygosity versus what might be expected from a large and completely outbred population. So obviously none of the world's populations are completely random outbred population. But the Taiwanese are the closest of any of these. And then what's shown on the sorry, that was the y-axis. Along the x-axis is the allele frequency, because of course the degree of homozygosity expected is a function of allele frequency, and the deviations are strongest at low allele frequencies. So you can see about 100 times higher degree of homozygosity in South Indians and individuals from Pakistan. So Dinesh has done a lot of studies of homozygous loss of function alleles, or has done studies and capitalized on the fact that there's a high rate of consanguinity in Pakistan. So it really made that kind of a something that's more generally recognized in terms of human genetic studies. But you can see here that South India has a very similar level of excess homozygosity, and the Bangladeshi and West Bengal individuals also have a pretty significant increase in homozygosity. So there's a number of reasons why this is important. I took this from a recent Nature a publication on the NOMAD project, and Beryl is, I think, in the audience, and she's an author on this. But it was highlighting the value, I guess, but more importantly, the kind of the sample size considerations for identifying loss of function genetic variation. And I think the point of, the A, B, C, D, and E is intuitive, that excess homozygosity that comes from consanguinity increases the probability of being able to identify homozygous loss of function, but they went through the numbers. And I'll point out that the consanguinous samples that are illustrated here are from the same Pakistani study population that we're describing here. So there's some comparison there. And it's the consanguinous subfraction of those Pakistani individuals that are highlighted here in terms of the sample size necessary to saturate all of the possible observations. So what our collaborator Jeff Wall did, Jeff at UCSF, was to use the whole genome data to model various patterns of parental relatedness, run that as a simulation, and then compare data from the whole genome data to that model. And so specifically what he looked at was runs of homozygosity above a threshold length after conversion to centimorgans, and then just look at the number of runs of homozygosity above that threshold. And if I'm remembering correctly, it was 10 centimorgans for the modeling. And there's, 5 different patterns patterns of parental relatedness that are graphed there. Unrelated, where you can't distinguish any kind of parental relatedness based on the model. First cousin once removed, second cousin, first cousin, and a avuncular. And I'll just point out that those descriptions are illustrative. So there's, in many cases, more than one possible parental relatedness, but these ones are easy for most of us to think about. So avuncular is uncle niece, for example, if you're not familiar with that. First cousin, I think, is straightforward, etc. So you can see the patterns that are present in the different subpopulations that we're characterizing, South India, Pakistan, and Bangla. And then what's plotted on the right-hand side is the degree of homozygosity, again, observed over expected, if one focuses only on the inbred, which in this case is the top 2 categories, avuncular and first cousin, everybody, which you saw before, the middle curves in each case, and then outbred, which is, quote, those that the parents do not have a discernible relationship from the model. And you can see that, and not surprisingly, the degree of homozygosity is really driven to a large degree by the amount of consanguinous unions, consanguinous parentage, but that there's a pretty, in the groups as a whole, there's still a significant amount of excess homozygosity. You can then characterize that with respect to the number of loss of function homozygous variants per individual, given how they're categorized into those different categories. So again, it's a more fine-grained description of the degree or the probability of finding homozygous loss of function alleles in an individual as it relates to the pattern of parental relatedness in a purely observational fashion, not a predicted fashion. And if you look at so now we're looking at runs of homozygosity that are greater than 1 centimorgan. So this is not the model. This is just looking at runs of homozygosity that are above a lower threshold. You can see there's samples from Han Chinese at the top. I apologize, the labels are different here. I need to harmonize the labels. BRB is samples from Bangla samples. There's Pakistani samples, and then there's 3 different groupings from South India: Coimbatore, Madras or Chennai, and Bangalore are the bottom 3. So the one thing is, of course, the 3 South Indian subgroupings look similar. They look similar to the Pakistani. And then if you take the Pakistani samples and take the inferred parental relatedness inferred from the genomes, you can see that the degree of homozygosity, that essentially you deconvolute the tail, the rightward tail in increased stretches of, longer stretches of homozygosity that you see in the plots on the left. So I want to come back to the founder effects, because that's the term that's been used to describe the this phenomena in previous studies, because it does approximate the same or the similar genetic drift that you see in founder populations that meet that semantic distinction. In this case, of course, intuitively, it's coming from the pattern of consanguinity and endogamy. So, we broke things down into, consanguinous, everybody, and unrelated parents in the previous, one of the previous plots. But what I want to highlight here is looking at those individuals whose parents are unrelated as terms of the modeling, the inferred parental relatedness. And you can see, again, with reference to individuals from Taiwan, which are similarly, filtered, that individuals from South India and Pakistan have even though they don't, in their parental generation, don't have consanguinity, there is the historical cultural patterns that you can that have contributed then to this excess homozygosity. So how much it breaks down between groups, et cetera, is a really interesting question, but one that's difficult to address without a much, much larger sample size. Once you start subgrouping these groups into smaller and smaller groups, obviously it's harder and harder to answer any of these questions. So I'm going to more or less wrap up here and turn it over to Eric. And so the segue is, again, coming back to the goals in terms of phase 2, producing tools, both a descriptionation of the population structure in terms of thinking about designing genetic studies, and then tools for carrying those out in an economical fashion, improved imputation and an optimized SNP array, and Eric's going to talk about those. There's some figures here in terms of performance. I'm not going to get into those, other than to say we see some pretty significant improvements in genotyping abilities by using the tools, and so we're in a good place for moving on to further efforts. So I'm going to try to stop and share if I can stop sharing. If I can, Fa, can you turn off my stop share? Because I can't find my cursor. Oh, wait. Here. Okay, there, I did it. Yeah.
All right.
So.
Okay, can you see my screen?
Yep.
Okay, great. Yeah, so yeah, I was just talking about this network that MedGenome is established across India, and it gives us access for doing medical genetics across India in terms of access to different types of patient cohorts. So really what we're trying to do is build up tools and technologies that will enable medical genetics across South Asia. So we have a large clinical exome database, mostly focused around rare disease. So it ties in genotype and phenotype data. We have about 50,000 samples. We have basically early access to the GenomeAsia data. We've committed to basically building out various disease programs in both in terms of genotyping and phenotyping. So about 25,000 samples over the next year. And then we built up a lot of data and analytics capabilities around doing familial studies. And then, so this is all on the data side. And we basically, in the US, we focus a lot on research services. So we've basically built up different technologies to apply in that space. I'll be talking about this array called SARGAM. But we do all the standard NGS types of sequencing, including exome, RNA-seq, single cell, and a lot of cancer-specific types of sequencing. And then on the data analytics side and software side, we have a lot in place. So we actually have a lot of expertise on human genetics analysis. We've actually developed a South Asian specific polygenic risk score. I'll touch on that briefly. And then we basically built up a lot of informatics capabilities around genomics. We have access to basically clinical interpretation of data. So on the diagnostic side, we have trained genome analysts. We built up a lot of software capabilities, so we can actually tap into that even for research projects, which is nice. We do a series of custom bioinformatics services work, And then we actually put in place a lot of software infrastructure. Actually, the origins of the founder of the company actually came from the software side of things. So we actually have a pretty strong software team. We've actually built up a lot of in-house software to basically power not only diagnostics, but research services as well. So yeah, so last fall, we announced that we were going to build the SARGAM array. We, as Andy mentioned, we partnered with Thermo Fisher. So Thermo Fisher has a lot of expertise in building these regional specific arrays. And so that's why we partnered with them to do that. And so what we wanted to do, where there's actually many applications that we thought of in terms of how we could use this array. One was essentially in the context of things like target discovery. So your standard types of GWAS, just using it in the context of quality control. It's still a really large, cost-effective way to generate, genotype data on large data sets. And sort of on the diagnostic side, we're interested in things like, South Asian specific polygenic risk scores, aneuploidy, potentially applying this in the context of pharmacogenomics. And then we actually have a DTC offering in India specifically we do things like ancestry and report other interesting traits. And then we could use it in the context of research services. So basically strategic collaborations where they may require genotyping of South Asian individuals. And so really this is the first array ever designed specifically for South Asian individuals. We basically used a series of in-house data, including GenomeAsia data. And then our goal was to basically get down to be able to compute, pretty accurately, even down to like 0.1%. And again, we partnered with Thermo Fisher, who's done other projects like UK Biobank, FinGen, and many more actually using the same technology. So why do we need a custom array? This is just one example of why. So basically, this is a comparison at that time to a couple other chips, one from AFI, one from Illumina. And what you're looking at are South Asian allele frequencies on the x-axis here and the number of markers here on the y. And it turns out that you basically get a lot of wasted space on an array. So these arrays are not optimized for South Asian individuals. So you're getting close to 150,000 to over 200,000 markers that you will never see in a South Asian individual. And the other piece, right, that's not on the slide is we had a chance really to optimize the array in terms of imputation and capturing of functional variants for South Asian individuals. So this really allowed us to kind of work with Thermo Fisher to design this from the ground up to really optimize this for South Asian individuals. So this is actually
Eric, can I ask a question?
Sure, please.
Yeah, I don't know much about genotyping arrays. Like, is the sense that for European populations or things like that, that basically the arrays are pretty well optimized, or is this just kind of generally a problem with
Yeah, so they have, obviously there's a lot of arrays on the market. People have been doing arrays for a very long time now. So they have a lot of different arrays depending on what the application is. They have one product from Illumina called the Global Screening Array, which is a very popular array basically where they're trying to capture as much diversity as they can. But the problem with these arrays is you have limited space. You can only put so many markers on there. And so there's no way to, do a deep dive on any one particular ethnicity. And so and no one's actually ever done this, for South Asian individuals specifically. But yeah, if you're there's lots of arrays if you're, going to other European populations that would work extremely well. Yeah.
So really optimization for the population is the advantage here.
Yeah.
All right.
Yeah.
Well, thanks.
So yeah, so basically this is what we ended up putting onto the final array here basically. So we've actually captured over 100,000 functional variants from different sources. We basically captured, direct to consumer markers, specific immunity markers, other tissue specific types of markers, a series of research markers. And I'll touch on that in just 1 second. We actually have a very large GWAS grid, and this has been completely optimized by the Thermo Fisher team, who basically specialize in this. So we basically provided them GenomeAsia data, and they're able to actually optimize our imputation grid specifically for South Asians. A number of disease markers are on the array. Again, this has all been optimized for South Asian individuals, and a number of pharmacogenomics markers as well. So just one example of one of the research projects we put on there. Actually, we have a paper in bioRxiv that we threw up right at the beginning of the pandemic because we all wanted to do something, and so we basically searched all our databases. We have access to a lot of internal databases from public sources as well as private data, including GenomeAsia data, and basically and since then we've actually done a lot of functional validation of some of these variants, but that's not included in the bioRxiv. But we're about to submit a version of that. But basically, we captured all these polymorphisms, so these rare ACE2 variation, and basically, at least in vitro, they're predicted to have some effect on binding from COVID actually. So we just happened to be putting up the final array right around when the pandemic happened essentially. So we actually tiled all these variants for ACE2 on that array. So we're calling it a SARS-CoV-2 relevant array because we're actually capturing that on the array, all these rare variations. So yeah, so as I mentioned, this is one of the more popular arrays to use in sort of a global setting. It's called the Global Screening Array from Illumina. This is some of this is from their website essentially in terms of performance across different populations. So SAS is the South Asian population there. It doesn't do quite as well as other populations, including Europeans. Actually, this is on their website. They highlight the fact that this array is being used by companies in India like MapMyGenome actually. So what we did actually just sort of a head-to-head comparison on a set of samples where we had whole genome data as the true set, and basically just compared, and we just wanted to compare apples to apples essentially. So we actually just picked out the sites that are captured, the markers that work on the Global Screening Array 3 and the markers that we have on our final SARGAM array, and just took the whole genome data and asked basically, if we use the same imputation panel, how do we do? And these are the results essentially. So this is basically the accuracy at different minor allele frequencies. And this is the GSA 3 on the left in blue and the SARGAM array on the right. And actually we do better in every category, especially at these lower allele frequencies. Actually, we get a huge boost in overall accuracy actually. And this is just using the 1,000 genomes as a reference panel actually. But overall, we see a huge improvement right away just with going with the SARGAM array over the Global Screening Array. And this is an independent data set. So this wasn't used as part of the design for SARGAM or for GSA because it's actually a private set to MedGenome. So yeah, so as Andy mentioned in his talk, basically, not only can we use GenomeAsia to help design these arrays, we can actually use it as a powerhouse for imputation essentially. So we actually have, this is the most high coverage whole genomes for South Asia ever assembled. So we can use that actually to basically boost our imputation as well. And so what you're looking at here basically is pulling up the markers from SARGAM, comparing it to GSA 3, but then we basically combine it with the GenomeAsia phase 2 imputation panel, and then just go with what essentially is the state of the art now, which is Global Screening Array 3,000 genomes of imputation panel. And we get even a further huge improvement actually, and this is, you're looking at different minor allele frequencies for South Asian individuals here on the x-axis. So we get huge improvement at these minor allele frequencies going with this the combination of the SARGAM array with the GenomeAsia phase 2 imputation panel actually. So so dramatic improvements for South Asian individuals. And we also looked, my slide is stuck here. I'm not sure, I have to, I'll just skip this slide, but basically we also compared to TopMed. We're actually outperforming TopMed just using the GenomeAsia phase 2 imputation panel across different parts of South Asia. So we also wanted to capture a lot of South Asian specific functional markers actually. So again, just as a comparison, we're just looking at comparing it SARGAM to GSA 3, and looking at this in the context of functional variants you would find in South Asia, right? So these things tend to have bigger effect sizes, right? Because they're protein altering in nature for other types of markers. And so from a medical genetics perspective, they become quite interesting. So basically start out with sort of the same number of markers between SARGAM and GSA 3. And here's just the ratio here basically. And then basically these are South Asian specific protein altering variations captured. So right away we get a boost actually. Or sorry, this is just protein altering in general, I guess. So we right away get a boost. And this is basically seen in South Asian samples here in either one of the public data sets, so NOMAD 1000 genomes or GenomeAsia phase 1, and it's damaging basically. And so here we get a dramatic boost, 5.5-fold increase in terms of number of functional variants captured. Here's basically sort of a little Venn here between the two, right? So we have 6,000 common markers, so we capture 6,000 of those 9,000 here. And basically a lot of the things that are still on here are pretty rare actually. So they didn't actually make it to our final array. So actually, if we look at just damaging or loss of function variation, we actually capture almost 75% of the genome, right? And this is the number of unique genes, so almost 15,000 unique genes, which is 3-fold over the GSA, and then about a 6-fold enrichment for loss of function alleles over GSA3. And this is the number of genes down here. So big advantage in terms of the number of loss of function or damaging missense alleles that we're capturing on the array. So these are just the numbers essentially that I talk about. So basically I mapped this onto what I call gene space. And so this is just a little grid. It actually represents, there's little squares there, hard to see. It represents 20,000 genes essentially. And I just kind of fix a gene, a little square there, and then kind of lock it in for the plot I'm about to show you. So what you're looking at basically is damaging missense or loss of functional alleles captured across gene space on SARGAM. So as I mentioned, we capture about 75% of all genes actually using the SARGAM array. And then this is basically looking at genes, that same gene space and the global screening array 3. So we have about 3 times more genes, and not only do we have more genes, we have about almost 6 times more alleles on average over GSA3. Again, this is all comparing for South Asian individuals actually. So if you're interested in medical genetics in South Asia, SARGAM has huge advantages. So yeah, so the use cases, I mentioned actually MedGenome has a DTC offering in India called MedGenome, which does ancestry and other types of reports. And that's just a new product that's just been launched by MedGenome. And so we plan to basically use the SARGAM array to power the MedGenome moving forward. We're interested in applying this in the context of polygenic risk scores. So this is, you're probably familiar with polygenic risk scores. It's been popularized by Sheikh Kadrisan and others in recent years, and that it may have clinical utility. Of course, the poster child for this is CAD and basically how you can intervene with therapeutics if you have a high polygenic risk score. So we've actually teamed up with the Broad Institute. So this is work with Amit Kara at the Broad, and this paper is just about to come out. This is directly from the proofs actually. So what we've done actually, what MedGenome has done in collaboration with Amit, is basically build a South Asian specific polygenic risk score for CAD. And so we basically we calculate a raw polygenic risk score, and then we can basically take PCs from South Asian individuals and then make an ancestry adjusted polygenic risk score actually. And the performance is pretty good actually for that. So but it's a polygenic risk score specifically for South Asians actually. So we're interested in expanding this in the future and using SARGAM actually to power this for additional polygenic risk score development. So yeah, so basically this is sort of the conclusion essentially about why SARGAM for South Asian medical genetics. And then I thought, since this is a bioinformatics meeting, I would just talk briefly about some of the software capabilities that we put in place at MedGenome. And so there's actually a lot of internal software products that have been developed that we use basically to power diagnostics, to power research services. So we have basically, so each one of these is different data sources essentially. So we have a pretty robust lens that we've developed actually. We have something called the patient information capture system, which captures different phenotypic characteristics for research projects. We've built a specific portal for capturing familial types of studies. We sort of normalize the phenotypes with this Phenomy system. And then on the diagnostics and interpretation side, we have programs called VariMat, which basically does custom genome annotation, pulling in all our different databases that we have internally to really, that gives us basically additional understanding, right, of interpreting South Asian specific variations and things like allele frequencies for South Asia that we find in our data sets, other pathogenic variants we reported out. We actually use machine learning to basically prioritize these variants, and that's all powered by these in-house data sources and annotated through this VariMat software. And then VarMiners are in-house, basically variant interpretation software. And this all goes into to basically a database that captures all this relationships between genotype and phenotype data. And then Mango is actually our analytics engine, essentially. So this is integrates directly with our LIMS. And I thought I'd just give a little bit of overview of how this works, actually. So we actually use a system called Nextflow, which is a pretty popular workflow engine you may have heard of, used in Europe. Actually, it's nice because with the same Nextflow script, you can easily and kind of readily deploy by just changing the configuration either on your local cluster. We use Slurm internally, but it supports basically any kind of scheduler, all the popular ones at least, or you can easily deploy that in the cloud as well. And then we basically built the Java system where around it. So basically web-based interactions on the front end, so you can easily just, anyone can actually launch analysis, and also basically for loading all these reports sort of on the back end as well. And then we basically, because we have multiple sites, we do everything. We build Docker images for everything and just can deploy that around the organization. So basically, it's set up where the bioinformatician can basically focus on writing, what they're using Nextflow to write an analytics scripts, and then just dropping it into the system from both. And then and the software engineers basically can take that to and basically create web-based interactions on the front end and back end, essentially. So yeah, so as I said, it's just all integrated with our LIMS. You just select the project, you select the samples you want, you select the parameters you want, and you click go, and it just runs essentially. And then to actually create this, you just have to do a runtime registration as a bioinformatician, actually. So you would write, as a computational biologist, you would just write your workflow, kind of make it work. You just drop it into the system, and then you just register the front end, say I need, and basically has all the sort of standard things you would expect, like drop-down menus, checkboxes, other text fields, etc. If you need to add a new species, you just click a button, add it in, and then it's basically, then it actually creates that custom front end for you from the get-go, actually, and then we just launch our analysis that way. And then we do the same thing for the reports on the back end. So your pipeline is generating text files, maybe HTML files, maybe some PDFs, etc. So we do the same thing. You can kind of move you can move things up or down. You can edit these. You can add new fields, whatever you want. So it's a one-time registration of your workflow on the back end, essentially. And you can do things like, add a HTML report and basically just give it the expected file name. The system, of course, knows where this file lives. So it's sort of organized in that sense. And even a text file, you can just drop in. It actually makes a nice interactive HTML table for you, actually. So you don't have to, you can take advantage of a lot of the engineering that's been done on the Mango system. So this is just basically, so what it is really just organizing all your, make a lot of interactive graphics, actually, that we can just drop into the system, actually. And yeah, so this is a TCR pipeline, and it has a various different reports, and becomes pretty straightforward, actually, to make pretty professional, nice looking outputs as a computational biologist without really having to focus on GUI stuff. I think, and because I think we're running a bit long, I think I'll just stop there, actually. And yeah, happy to take any questions. These are just acknowledgements from MedGenome and Thermo Fisher, who we worked with closely to design SARGAM. So yeah, thanks everyone.